avian influenza are all the avian strains of influenza A virus. When we go out and we do our surveillance for avian influenza, we actually find an enormous diversity of these H5N1 combinations. However, when we generally do our surveillance in Australia and overseas, what we find are low pathogenicity avian influenza viruses. And so how did we get here? So it was the year 1996, and we had one of these low pathogenicity H5N1 viruses, so an H5N1 that doesn't cause disease, in poultry, and this virus evolved. And so what we ended up with was a virus that transitioned from one that didn't really cause disease to one that really caused disease. These high path H5 viruses became endemic in poultry in some parts of the world. And as they circulated in poultry, they were never eradicated. And with that comes evolution. And so the same way we saw in the COVID pandemic that all these different variants emerged, we now have a huge number of variants that have emerged of this H5N1 high pathogenicity avian influenza. So the variant we're here to talk about today is this one called 2344B. So before the emergence of this 2344B variant, if we look at what the epidemiology of these other H5N1 variants, what we generally found was a virus that sticks to chickens like glue. It's endemic in chickens, it would circulate in chickens. In poultry that were vaccinated, the chickens wouldn't get sick, and so it just got sustained. And these viruses almost never jumped out of chickens into wild birds. And the few occasions that they did, it ended up being self-limiting, so it just stopped. But when this 2344B variant emerged, the whole situation changed. So now we have a virus that does well in chickens. We've got a virus that is reservoir in wild birds, one that's a reservoir in some mammals, including seals and cows, and from there spilling over into a huge diversity of other animals, which has really led us to the global pandemic or animal pandemic that we're in.